I work in a cancer research group at Biomedic. And my topic for today is learning-based detection of early neoplastic changes in histological images. So first, a little bit of motivation. So prostate cancer is the most common male malignancy in Western countries. Prostate cancer is kind of multifocal cancer, which means that it can develop lesions that are slow growing, and then again it can cause really nasty and potentially lethal lesions. So it would be really important to detect these changes as early as possible. So to understand this multifocal prostate cancer, we developed some histological tools to analyze these host-like images and studied early neoplastic changes in mouse prostate. So we developed this image analysis pipeline for histological samples. First, the mouse prostate tissue samples are sectioned. Then these sections are stained with hematoxylin and eosin and scanned into high-resolution host-like images. Then some regions of interest are segmented by an expert. Then we did some multiple image analysis steps and extracted multidimensional feature data, and finally used machine learning to analyze these regions of interest. So in this study we used P10 mice. These mice are lacking the other allele of tumor suppressor P10, which means that at 10 to 11 months, all these mice will have lesions of intraepithelial neoplasia in the prostate. These PIN lesions are considered to be an early pathological change preceding cancer. So as the name implies, these epithelial cells grow only inside the lumen of these PIN lesions. Here's an example of whole tissue section of mouse prostate. The tissue is very heterogeneous. There are like nerve bundles, stroma, muscle, and also these different types of normal prostate tissue: lateral prostate, ventral, and dorsal prostate. Sorry, hard words. So let's look more closely to the different sides of the tissue, so this lateral, ventral, and dorsal. For example, if you look at this lateral prostate, it would be quite easy to detect this epithelial cell growth, as the epithelial cells are arranged in this really thin row. However, when you look at, for example, this dorsal prostate, the epithelial cells are already growing quite thick inside the lumen. So to detect these early changes from all these different sides, we analyzed this expert segmented dataset. In this dataset, we had normal tissue samples and these PIN lesion samples from all the different sides: lateral, ventral, and dorsal. In total, we had 244 normal samples and 198 PIN lesion samples. So here are the main image processing steps that we used. We did histogram matching, color deconvolution, and nuclei segmentation. Histogram matching was applied to correct the color variation across the tissue samples. We selected few samples that had visually good stains and then generated a target histogram. We computed a mapping function, and then for each sample we matched a histogram to the target histogram. Then we used color deconvolution algorithm to separate the hematoxylin and eosin stains, and segmented cell nuclei. For this we used the hematoxylin channel, as it mainly stains the nuclei. So first we did some Gaussian filtering, then thresholding to get the binary image, and finally we used Watershed segmentation to separate these overlapping and touching nuclei. Here is an example of the segmentation result. The next step was this feature extraction. So the main idea, of course, is to get a numerical representation of the tissue samples. The features were extracted from all the channels, hematoxylin and eosin, and then also from the segmented nuclei. The features that we extracted included local descriptors related to tissue texture and spatial arrangement and distribution of the nuclei. In total, we extracted over 700 features, but we used approximately 100 features for this analysis. I will shortly show you just a couple of features that turned out to be important for detecting these early changes. So first, these texture features. These were calculated from the nuclei area and nuclei neighborhood. These included properties from gray level occurrence metrics, such as contrast, correlation, and energy. Here you can see a set of sample plots from where these properties were calculated. We also extracted histogram properties like skewness and kurtosis. These samples are from both PIN lesions and these normal samples, and you can see there's the, not very well, but the hematoxylin and eosin stains. Then we extracted these nuclei distribution features. These features were extracted from a bit bigger neighborhood, which has the nuclei locations marked. Maybe you can see them, but yeah, we calculated, for example, distance features and directional statistics from these. So, for example, the angles between the center nuclei and to the other nuclei, and you can see in the polar plot those angles. So, and when you look at this angular histogram, the difference in the arrangement of the nuclei between PIN lesion and normal tissue is quite visible. So, in the other one, they're like evenly spread, and in the normal sample, you see that there is only there is a skew in the histogram. So, you can see that they're arranged in this thin row. So, the future representation of tissue samples were then used to train a random forest model. Our random forest model was an ensemble of 50 decision trees, and it uses bootstrap aggregation to improve stability and accuracy. So basically what the model does, it estimates the probability to belong to the group with neoplastic changes. For validation, we used Lee-1 outcross validation. So we trained a model for each sample that was trained with every other samples. Then the results. So our model was able to separate the normal samples from these PIN lesion samples. These figures are from all the sides. So the lateral, ventral, and dorsal are combined together. For example, up on the left, you can see the confidence values that our model gave for the samples for belonging to the early neoplastic changes group. PIN lesions obviously got higher confidence values, and these normal samples got much lower confidence values. Also, our model gives this information about which features were most significant in the classification. You can see there, for example, those distance features and directional features. We also calculated this receiver characteristic curve, which illustrates the performance of our classifier. And area under the curve was also calculated and got quite good results, 0.97. Then there's also presented this DSNE analysis of the feature data in a scatter plot. visualize that you can actually separate these two groups. And then when you look at this TSNE analysis for only these normal samples and then only for these thin lesion samples, you can see that normal samples, the different sides are actually separable, but however in this thin lesion scatter plot, you can't separate these anymore. So spatial heterogeneity in the tissue samples decreases as these early neoplastic changes occur. I have also these confidence values and ROC curves for all these different sides. So quickly, in this lateral prostate, which is basically visually easiest to separate these thin lesions, it gave really good results for ventral prostate also. And even for this dorsal prostate, where it's not visually so easy to separate these normal tissue glands from these early changes. But yeah, our model was able still to separate these two groups. And to conclude, quantitative analysis of early changes and spatial heterogeneity of prostate tissue samples was presented here. This easily interpretable model was able to discriminate normal tissue samples from these early mouse pin lesions. The feature significance of the model provides a short list of features potentially linked with neoplastic changes. And well, this method actually generalizes to different tissue types. For example, metastasis detection from whole slide images of lymph node sections. We actually participated in this grand challenge Wanderlog already mentioned. We used the same method as I presented here, with just small modifications. Actually, we're the best team that used traditional machine learning methods. So, yeah. And quickly about future goals. So, now that we have these tools to extract numerical information from these tissue sections, we are working on combining this with 3D growth patterns. Here's a good example, for example, the limits of 2D images. You can see there is, from these 2D images, you can see that there are actually 2 lesions on the other image and 1 in the other, so these are really important features that 3D models would bring. And finally, here are the people that have contributed to this work. So, thank you. Yeah, thank you.